So it's great pleasure, Danny, to introduce Dr. Sabin. He is at Duke University, where he's an associate professor with tenure by way of University of Florida and then at Harvard. And he worked with some great people while he was training, Maria Grant and Wayne Stryline. Danny, you may not know this, but Wayne was on my thesis committee. So it's like 2 degrees of separation, you know. So Danny has done fantastic immunology throughout his career, working both on innate and adaptive immunity, and most recently on microglia, which is what he's going to talk to us about. But first of all, I mean, if you just PubMed him, you'll see how prolific he is, and it's a little bit, it's impressive and a little bit daunting. And so the diversity of publications in terms of either T cells, monocytes, microglia, and I'm really excited today that he's going to talk to us about his most recent work on retinal niches and microglia and how they're important in homeostasis and disease. The only thing I mentioned earlier that I'm a little disappointed about is the best way to listen to Danny usually is you know, to hear him talk about science when you're having a drink with him. So I'm sorry we're not able to do that, but maybe in the future. So Danny, I'll let you take it from here.
Well, I see. Thank you for the kind introduction, and I see Brian Edelson is on. And if anybody wants to know what I'm like talking science and drinking, maybe Brian can fill you in. Thanks for coming, Brian. Good to see you.
Good to see you.
And that was a very warm introduction, Raj. And I feel humbled. And I think you're talking about my publication stuff. You could publish yours, and yours is very, very impressive. And I see you just published something recently on COVID. So anyways, this is an honor to be here. So thank you for the invitation. I look forward to giving my seminar, getting some feedback, and of course, meeting with some of the folks that I get to meet with. Okay, so today I'm going to talk about microglia, obviously, and how they function in a sort of anatomic or subanatomic or microenvironmental fashion. And being in ophthalmology my whole career, essentially, and studying immunology my whole career in the eye, the retina is quite a fascinating place to study this. And hopefully those of you that are not looking at the eye can appreciate some of the facets about the retina in terms of its utility and looking at unique models and synaptic interactions and mapping out immune responses, etc. So hopefully I can pass that enthusiasm along. Let's see here. Let me just move this again to a pointer. Laser pointer, and next slide. Okay. So I would be remiss if I didn't speak about macrophages in general. Microglia, as you know, are macrophages. And I think in the last 10 years, there's been an explosion in terms of what macrophages are, what they do, what their ontogeny is, and lots of you out there know about this. I'm standing on the shoulders of giants in understanding this, and I think with that we've started to appreciate even more so their role in just normal tissue physiology, as opposed to just thinking of them in the context of immune responses and protection from evading microbes, etc. So here's a paper, a review written by, or a perspective written by Akabi and Ruslan Metsitov, who I'm sure most of you know of, just highlighting, these macrophages in the bone marrow, for example, osteoclasts involved in bone resorption and remodeling. Of course, you know a lot about this alveolar macrophage, which is important in clearing surfactant. So it helps in surfactant homeostasis in lungs, which of course is critical in breathing and oxygenation. And then finally, here's another example, which will bring us to our talk, or my talk here, which is microglia involved in synaptic pruning. We know of many people's work, including Ben Barris and Beth Stevens, of course, for the role of microglia in helping prune and establish these circuits. And of course, we can't move forward without discussing this breakthrough paper. This was in 2010 by Ginu and Murad and colleagues, which essentially established microglia as yolk sac-derived cells that come from the yolk sac and maintain themselves in adulthood as microglia without significant input from circulating monocytes. And that had changed sort of the paradigm from the Van Furth model, which was essentially that circulating monocytes continually make up the pool of macrophages in all tissues of the body, or at least most. And of course, there were questions about some of those. There was emerging findings in the brain. For example, microglia sort of were long-lived. We're being able to self-renew to some extent. In Langerhans cells, they were showing that in skin. But nobody had really generated a paradigm shift like this paper itself, I think. And this is just an adaptation from a great review from Ginu and Guillaume's in 2016, essentially showing what had happened after that paper. We started looking, not me, but the field started looking at macrophages in all the different tissues and found out, well, there's these long-lived self-renewing macrophages in most organs and in many tissues. So if you think about the long-lived, what called resident, of course you have them in the brain, the microglia, but also in the border-associated macrophages too, perivascular macrophages, for example. I gave you the example of alveolar macrophages. Those are also long-lived, self-renewing. Of course, the epidermis and the Kupffer cells, which is a lot of what Guillaume's is working on. And at the same time, there was still some truth to the Van Furth model, probably a lot of truth, in that there are monocytes in these open niche tissues, as Guillaume's has suggested, where you have, for example, in the lamina propria of the gut and in the dermis, and to some extent in the pancreas and heart, you have monocyte-derived cells in adulthood. So initially you have the seeding and these macrophages, long-lived macrophages that are established, but eventually they give way to bone marrow recruits. And these are called open niches. This is different than closed niches, for example, in the brain, where you have blood-brain barrier, or in the alveoli, where you're not exactly in the tissue itself, or the Langerhans cells, where you're in the epidermis, where there's less vascularity. Now, the other interesting thing is once we started to appreciate the difference between these residents versus recruits, or they have been called passengers, for example, then we began to realize that these, in the inflammatory setting, when you have monocyte recruitment coming in, that these populations, when you're looking at organs and tissues where you have long-lived self-renewed, that you essentially have two populations in an inflamed state. You have these resident transient wave of recruits, potentially maybe transient in most settings, but maybe not if you have a chronic inflammatory setting. And then you have the resident cells. So the question now that emerged is, you have these two populations with different ontogenies. Is it possible that they're functionally non-redundant? And most of what we're learning is that they have a lot of unique specializations. And of course, there are some redundancies. Now, you can even go even further and ask, so if you're looking at microglia themselves, are there differences in different regions or some anatomical regions? And certainly, if you look at the morphological evidence, for example, in this paper by Lawson in 1990, certainly you can argue that there may be differences, right? If you look morphologically. But of course, you can counter-argue that point and say, maybe the shape of these cells is really just structured based on their cellular environment. For example, in the corpus callosum, where you have these long type microglia, that's because you're along axonal tracts. Whereas when you look at the hypothalamus, where you see these highly decorated cells, this might just be because they're interacting with synapses and there are other cellular components there that they're interdigitating. So you can argue it both ways. Now, in 2012, you had two papers, one that came out of Marco Colonna's lab, I believe, clearly out of WashU, and the other one, Burkhard Becker with Miriam Murad and such, essentially showing the same thing, which is that IL-34 is quite critical in maintaining microglia. But if you look closely, you can see, sure, there are many brain regions that do have need the microglia maintenance through IL-34, such as the hippocampus. But then you can look at the cerebellum, for example, where IL-34 is not needed, or the brainstem. And even within these structures, you can see that approximately half of these microglia are IL-34 dependent and some aren't. So this, again, builds on the argument that possibly there are some differences in microglia based on region or even subanatomical space. And even more evidence pointing toward that as a possibility. You can see this really nice paper by Grabert et al., where they use, now we're in the NGS next gen period of time in science here, where you can pull out microglia from these different locations and then do transcriptome analysis and use principal component analysis to show that indeed they cluster quite uniquely based on anatomical or brain regions. So that's more evidence that these microglia could be different based on where they are. Again, but you know, all of these are really just descriptive type of reports. We really didn't at the time know anything at the functional level. And of course you can ask yourself, why is this even important? Why do we care? And you know, many can argue that it's important not only as a basic science question, but potentially therapeutically, or to understand pathobiology in specific brain regions. Clearly, Alzheimer's is one where you have microglia that surround the plaque. Marco Colonna showed that beautifully with Idomit and others. And so perhaps understanding those functions in a niche specific fashion will help in terms of treatment. And that's seemingly the case, I think, in the direction of the TREM2 studies. So I do think that this is an important question. Now, we've pursued this in the context of the retina. And as I mentioned to you, we're in an ophthalmology department, and there's a lot of benefits towards studying the retina. There's a simplification and there's a highly stratified anatomical layout, which I'll show you in a second. Of course, this is a bona fide part of the CNS. This is from the optic nerve, which is extension from the LGN, and SC from the brain. Now, what's quite beautiful about the retina in terms of microglia, if you look here, you have the structure of the eye. I'm just showing you DAPI staining. You can see the two different layers, or the three layers. It's the retinal ganglion cell layer, which then synapse with bipolar cells. Of course, there are other accessory neurons in here, which I'm not well versed on. But the main players here are these bipolar interneurons, which synapse with the RGCs, and then also synapse with the photoreceptor cells. So this is where the photoreceptor layer is in terms of their cell bodies. These are ciliated cells that, of course, receive light. They also interact, and this is a critical component. I won't focus on it right now, but later they interact with retinal pigment epithelial cells, which establish the outer retinal border from systemic circulation in the choriocapillaris, which is the next structure right behind it. And I'll talk about that later. But what you can see clearly is that you have microglia in these two layers. Of course, they're also in this nerve fiber layer, but we won't focus on that for today's talk. And they're both in synaptic regions. So this is in of itself very interesting. Why are microglia living in the synaptic regions only in the retina? That I will only touch on, but suffice it to say that you have microglia in the same tissue, which is the retina, but in two distinct layers, synaptic layers, which have have differing environments. So you can ask maybe with this system whether there's some distinctions between these microglia at a functional level. So as I introduced, we knew that microglia are long-lived, but they're also yolk sac derived. So we first wanted to establish, we didn't know in the retina whether this was the case. You can assume it, but it needs to be tested. So we teamed up with Florent Chinou over at A-Star, who uses or has the showed with the RUNX1 Cre-ER mice. These mice allow you to label yolk sac progenitors in the blood islands at E7.5, E7, E7.2.5, E7.5. And then you can look at the adult mice from the pulsed dams and ask whether the microglia there are YFP positive, essentially lineage tracing them back to the erythromyeloid progenitor cells, which are yolk sac derived from that period. And that is indeed turns out to be the case. So if you look at the retina, these are adult mice, and you gate on microglia, you can see that there's a fraction, a very nice fraction that is YFP positive. And you can see the percent recombination, which is slightly higher than the brain, but I don't know that I would make anything of that. I think there's probably in the brain, there's probably some meningeal tissue or whatnot that contribute to lowering that. But suffice it to say, we can say that these are yolk sac derived. Now, you not only have to be yolk sac derived, but to be a microglia, you have to be a canonical microglia, you have to be long lived. So to answer that question, we used the CX3CR1 Cre-ER mice. These were from Stefan Jung's lab, I believe. Of course, Dan Littman established these mice independently as well. I think these are the mice from Simon Yona. And they express the Cre-ER under the expression of CX3CR1, which is the fractalkine receptor. As many of you know, it's broadly expressed on myeloid cells at some points in a particular cell's development. It's maintained expression on circulating monocytes, and it's very highly expressed in microglia. So if you come in and you have a reporter and you pulse with tamoxifen, you're going to flox out the stop codon and have a reporter, whether it's tomato, etc., on both of these cell types, both the adult monocyte-derived and microglia. However, because we know that monocytes are short-lived, they essentially get washed out. So all those reporter positive cells get washed out. Whereas in contrast, microglia, because they're long-lived, they maintain the reporter positive floxed allele. Even if they divide, they can self-renew, the daughter cells should also maintain that flox allele. So essentially now you have a way of discerning these two. And this is important, of course, because monocytes become macrophages when they get recruited, and essentially phenotypically look exactly the same when you're talking CD45 or CD11B, even CX3CR1. And I'll show you that later on in the system. But this is probably the best way we know how right now, most validated, most widely used way to discern microglia from short-lived monocyte-derived macrophages. So you can do this in the mouse, which we're doing here. And we've pulsed now an adult mice about 8 weeks old. The reporter is GFP. IBA is a macrophage marker to a good extent. And we looked at this 12 months after pulse. So this is a year. So anything that is FGF positive, that's double IBA positive as well, essentially has lived for a year, which I think it's safe to say that's long-lived. So if you look at the retina, you can see both in the plexiform layers, all pretty much the microglia are FGF positive. So close to 90 or so % are long-lived, which is what we expected. In the optic nerve, interestingly, and we're kind of slowly looking at this, we didn't expect this, but only 50% or so are long-lived. Now, the optic nerve is bona fide CNS tissue. So why they have short-lived cells there, we still don't know. And we're kind of slowly addressing this question with Roman Cartoni over at Duke. So that was somewhat surprising. You have the iris-ciliary body. That was a little bit surprising to us too. You have both long-lived and short-lived. Again, this mixed mixed macrophage situation here. And this is not bona fide, this is not CNS tissue, and there are some fenestrated capillaries here too, so we're somewhat surprised by this. And we're now looking at the role of macrophages in glaucoma with Katie Liu, who has a K award to study this, and it's a very interesting story. It's going to take us a little bit of time to get out, but it turns out that the macrophages are involved in outflow homeostasis. Now, if you look at the choroid is the support structure of the retina. I think you can think somewhat, to some extent, that the choroid is synonymous with the meninges. There's a lot of differences, but just, for those of you that are studying the brain, excuse me. But what's interesting here is we found no long-lived macrophages. They're all the IBA1 positive, CX3CR1 positive don't maintain the floxed allele after a year. So they're short-lived, or we can say at least that they're not long-lived. And that we've repeated several times. Similarly, if you look at the cornea, this is also interesting because the cornea is not vascularized. The cornea proper is not vascularized, yet the cornea proper, which is right here where you see essentially blank in terms of FGFP positive, there are no FGFP long-lived cells. So this is essentially filled with short-lived cells. And you can just show this by flow cytometry, just really quickly. This is FGFP versus CD11B. These are gated on macrophages. These are the different tissues I just showed you, the no TAM control plus TAM. So you can see in the retina, the majority of them are long-lived. This is 1 year after pulsing. We're just showing you that 48 hours, and then at 1 year later, most of them are FGFP positive or long-lived. The optic nerve, you can see there's a population that are, and there's a population that are not, which is shown here, about 50%. Iris ciliary body, you can see some are long-lived and some aren't. Same situation. And then the choroid, as I showed you, there are some CX3CR1 positive cells, but if you look after a year, they pretty much fall below threshold, so they're not long-lived. So there's a different profile in the eye. I don't know that people have studied one organ in terms of macrophages as completely as we have. But this is the case. There's different, and this is interesting to study, and we are studying that. Now, the other thing you can do now with these data, with bioinformatic platforms that are now widely available, here's a T-SNE, T-stochastic neighborhood embedding analysis, which most of you probably know. Essentially, it's just compressing the flow cytometry data. Here, we're looking at 10 or 11 markers into a two-dimensional space. We've asked whether the long-lived versus short-lived cluster differently or position differently on a T-SNE map, and they do indeed. So if you agree that all long-lived are lineage distinct from short-lived, then you can see that this lineage distinction actually clusters out. So the long-lived seem to cluster, and this is statistically significant, versus the short-lived in all of these tissues. This is not by tissue. This is just completely pooling the data together, irrespective of where you are in the tissue. If you're long-lived, you cluster. If you're short-lived, you cluster differently, suggesting that ontogeny, and we know this by now, ontogeny has an impact on the expression profile of these cells, and therefore you can probably extrapolate that there's some functional differences. However, at the same time, you can do this experiment in a different way. Now you can ask, if you look at long-lived and you separate them by tissue, do they cluster differently? And the answer is yes. So only long-lived macrophages in the optic nerve have a different expression profile than the ones in the retina, and a different expression profile than the ones in the iris, suggesting that there are tissue imprinting impacts of the expression potentially. So if you need an answer to the age-old question of nature versus nurture, I think the eye gives you that answer. It's both nature and nurture that seems to be important in life itself. That's just a joke. But essentially, that's what what these data suggest. Now, you go even closer, you zoom in, And again, as I promised, we'll look at these retina in a layer-specific fashion. This is in collaboration with Miriam Murad. What we found using these IL-34 reporter mice, this has a LAC-Z replacing the gene. The heterozygous mice have one functional copy of IL-34. So you can see they have microglia in both of these layers. What we were completely fascinated by and quite surprised was that there was only a selective loss of microglia in the IL-34 depleted setting. These are two LAC-Z genes. Only in the IPL did we see a numerical or abundance, a loss in these microglia. You can see these better in flat mounts. So this is the outer plexiform layer, relatively similar quantitatively. And this is the inner plexiform layer. You can see quite different, almost a 50% loss. So there's a selective IL-34 dependent niche in the inner plexiform layer, which was really interesting. This is the first time that this was shown. And this made sense because when we looked at sort of a closer, and this was done in collaboration with Jeremy Kay at Duke, we found that the ganglion cells were actually expressing the IL-34. And of course, the ganglion cells are the neighbor cells to the inner plexiform layer. Here you can see a brain 3A staining, and you can see these are perinuclear around these ganglion cells. And you can also look at this ATOS7 or MATH5 mouse. In the heterozygous, they have normal, relatively normal ganglion cell development. And you can see this is in situ RNA scope for IL-34, all the signal here in the RGC layer. And you can compare that to the ATOS7 knockouts, which don't have normal development of the ganglion cell layer. And you can see the signal is quite lower. Again, suggesting that the ganglion cells are nourishing or providing the IL-34 to these cells. Now, okay, so if you think about sort of these niche specific macrophages often have the systems, it's often a system, right? In the sense, if you think about the alveolar macrophages, it's the alveolar macrophages, the alveolar epithelial cells are providing I think it's CSF2 to help nourish the macrophages and determine their fate and function. And then the macrophages in turn are eating up the surfactant to help with homeostasis. So there's sort of a symbiotic relationship. And it turns out that's the same thing there in the IPL to some extent. Of course, we don't have this fully worked out. And I don't think that I have the expertise to work this out as an immunologist. But we're always open to collaboration if anybody's interested. But if you take these IL-34 knockouts, again, they have an inner plexiform specific loss in microglia. So their outer plexiform completely normal. So that really lets you like look pretty cleanly at synaptic interactions or information. And we have the electroretinogram, which is an in vivo electrophysiological assay, and it's really widely used in the clinic, of course, and widely used in retina research, vision research. And so you have this scotopic wave. You basically dark adapt these or these mice, you pulse them with light in the dome, and then you have these electroretinogram recordings. And in the A wave, we saw no difference. The A wave is indicative of essentially photoreceptor function and to this light stimuli, and we saw no difference, which sort of made sense because we didn't see any defects in the photoreceptors and outer plexiform layer, which is a synapses that provide information back and forth to some extent to the photoreceptors. The microglia were fine. In the inner plexiform layer, that was where the difference is. So if you look at the scotopic B wave, which gives you information more about the, to some extent, the bipolar cells, you could see at these higher flash intensity. In the low flash intensities, no difference. The high flash intensities, you begin to see a collapse of this ERG function. And at these higher waves, at these higher light stimuli, it's more cone bipolar cell, it's thought to be anyways, or shown to be, that are playing a role in these high intensity light responses. And you can see that's where the defect arises. And this is just showing that a little bit more. So you get approximately a 50% loss in bipolar cell or B cell responses at these high light higher light intensity. So I make the joke is that you can thank your microglia for being able to see during the day, because of course cones, among one of their jobs is to basically allow you to see or respond to light during the day. So clearly the microglia and the anaplexiform layer are playing a role there. So just to wrap up this part, this half essentially of the talk is, we see these two microglial layers, and we show that in the interplexiform layer, you have a microglia IL-34 dependent niche, whereby the ganglion cells are producing IL-34 that maintain microglia. And in return, microglia seem to be playing a role in helping at least the responses to light stimulus in the ERG, presumably to cone bipolar cells. But again, that's probably needs to be worked out, and exact mechanisms we don't know. We do know that there's no synaptic differences in terms of numerical, so it may be a qualitative issue. Again, that's not something we've looked into deeply. And then of course, in contrast, you have the outer plexiform layer, IL-34 independent niche, where microglia reside. And we don't know exactly what they're doing there specifically, but they are clearly independent of their need to be maintained of IL-34. So now let me sort of switch gears slightly to another layer. And this is what I call a de novo disease-associated immune niche. Normally, you don't have any immune cells here in the outer retina, where between the photoreceptors and the RPE. However, in retinal degenerative diseases, and you see this across the gamut, photoreceptor diseases, photoreceptor degeneration, retinitis pigmentosa, you see it in LCA. Essentially, you see it in congenital forms of retinal degeneration. You also see it in adult forms of retinal degeneration, and you also see it in age-related forms of retinal degeneration. That is an accumulation of macrophages in this subretinal space where there never was macrophages before. Now, it's not surprising, because in these disease forms, it's the photoreceptors that are dying. So clearly, it's not a surprise that macrophages are moving there, because you think they would phagocytose the dead and dying cells, and that's been known. Although whether they're microglia or not, now with these new definitions, that was never known. But what also wasn't really understood is, sure, okay, these microglia or macrophages are moving to this space to actually clear the debris, but are they also doing something bad to the tissue at the same time? For example, releasing cytotoxic molecules such as TNF-alpha that might cause collateral damage and further the problem. So it's not really only doing good. And the fact that they're in this critical place between the photoreceptor and the RPE also is food for thought, because you need interactions between these two cells for the photoreceptors to survive. These RPE cells are constantly on a daily circadian rhythm, actually phagocytosing. These are phagocytes themselves. The outer segment tips of photoreceptors that are basically spent and cleared. And they're also providing nutrients to the photoreceptors. And there's also some feedback from the photoreceptors to the RPE by way of metabolism. So these two cell types are really engaged. And so when you break that interaction with phagocytes, perhaps that is also detrimental to the tissue. So these were questions that remain unsolved that we thought to dive in. So this is what I'm going to focus on. Now, this comes back to the CX3CR1 CRE-ER. Just a quick reminder, you pulse the mice, both monocytes and microglia become reporter positive, or whatever gene that you're floxing out in that transgene or locus. And you can thereby separate, because the monocytes are short-lived, you can specifically target long-lived, in this case, microglia. So just a quick overview of the system. Again, in the inflammatory context, we're using the light degeneration, light toxicity model. This was established in 1966. It's an old model. Essentially, you're exposing mice to really bright light. so much so that it's causing apoptosis of photoreceptors. And it's really nice because it is tunable. You can crank up the light, or you can basically dose escalate or reduce. So if you tamoxifen pulse these mice, this is looking only at YFP cells in the blood. These are the monocytes. You can see they're all RFP. This is tomato. And in the retina, it's the same thing. These are the microglia. This is steady state. If you wait, you don't have to wait 60 days. You can wait about 2 weeks. Of course, you'll get the non-classical still kind of roaming in the blood. Here we're showing 60 days post-tamoxifen. Of course, now the monocytes have purged themselves. The ones that were TD tomato, they're gone. They've washed out. Now you only have YFP, but the microglia remain. And then you can take these mice and then induce a light damage, which causes recruitment. And you can clearly distinguish the two populations, the microglia, which are lineage traced RFP, or TD tomato in this case, and the recruits that are not. And you can now use this system to look at the tissue. So if you were just to use CX3CR1, this is the light damage system. You can see here's the outer nuclear layer where the photoreceptor soma are, cell bodies. And in the light damage setting, you clearly see it's thinner. And now you see this recruitment of these macrophages to the subretinal space, the thinning here. Now think about it. If you were just looking at CX3CR1, you wouldn't be able to determine what's what. What is a macrophage recruited monocyte-derived versus a microglia? The same thing goes for CD45 or CD11B, IBA1, F480, what have you. Now look what happens when you lineage trace. You see a completely different story. So this is in the steady state. Of course, the microglia are in the plexiform layers, and they co-localize with the YFP. In light damage, you find that the tomato-positive lineage traced microglia are in the subretinal space. So you have green everywhere, meaning you have recruitment of monocyte-derived macrophages into the parenchyma, but only the microglia seem to be restricted to this subretinal space, and that's shown here quite nicely. And you can take the RPE, the retinal pigment epithelium, flip it over to look at the apical side, and then probe with a microscope. And what you see is that the only cells that stick, and we've known for a while in this system, you get the macrophages actually stick to the apical side of the RPE. Nobody knew which cells those were, whether they're monocyte-derived cells or microglia. Here we're showing that they're completely 100%, at least in this image, and in most of the settings that we've looked at, are microglia. They're all lineage traced. If you look at the neuroretina, you can see that many green cells, but very few of them are microglia. So essentially in the injured region, microglia are all moving to the subretinal space in the injured region, whereas the monocytes are pouring into the tissue, and you only get restricted access to the subretinal space if you're a microglia. Now you can argue, well, this is a subretinal space and could be a lot of autofluorescence. These are phagocytes, so maybe you're just seeing and of course we did all the controls, but sometimes with autofluorescence, it's really hard to control for it. You do your best. So we also did the DTR system. So instead of the tomato, we used the DTR. And then we can use this DTR quite well, either to lineage trace with an antibody against DTR, which works beautifully, or we can deplete selectively these microglia. So you can see here in the light damage, we get the same thing. These DTR positive microglia are in the subretinal space. And what we expected, you deplete, you get no microglia, you're sparing the monocyte-derived cells. So you are getting still recruitment, but you don't have any microglia. But what was fascinating from this experiment is that clearly you see no microglia in the subretinal space, which is what we hypothesized. But what was interesting is that even in the absence of microglia, the monocyte-derived cells still did not go to the subretinal space. So it wasn't really it wasn't a competition or a race for who gets to this space. It's again suggesting that it's a unique microglia access to the subretinal space. And you can see some similar, potentially similar parallels of unique microglia access in the brain. For example, you can argue that around the plaques, which is also a disease-associated niche in neurodegenerative diseases like Alzheimer's, and it's been shown that really you only get microglia there, even if you've done, and I can't tell you the references of the papers. But even if clearly Ido Amit has worked on this with Marco Colonna, but really it seems to be restricted to microglia. And there's also a nice paper showing that in the choroidal plexus on the apical side of that, you have a restricted space that only microglia are. They're actually microglia, not macrophages. And just to show you here, you can deplete microglia, and you look at the apical side of the flat mounts, these RAP flat mounts, and you see no microglia there, which is consistent with the finding. And you know, the light damage model is highly artificial. You can use a more clinically relevant model. This is ROP23H. This is a knock-in. This was developed by Chris Palczewski. It's a knock-in mutation of a rhodopsin motif. They get degeneration, and this is a heterozygous, so the degeneration is somewhat slower, and you can see, of course, the accumulation of these IBA1 positive out to P60, even in P30, and you can see the thinning of the ONL. This is a slower, more progressive system or degeneration in the heterozygous setting. And we bred the CX3CR1 IDTR system onto it so that we can fate map and of course deplete, and we see the same thing. So if you look at the subretinal space, all of these microglia that are apical are DTR positive, meaning that it's microglia, not a monocyte-derived cell. And then you can deplete microglia and then show that you don't have any of these cells, or a very limited number of these cells, on the apical side. All of this proving that this is a phenomenon where microglia have unique access, both in an artificial model and a more conventional clinically related model. And we went one step further. So now we use single cell RNA sequencing. And I have to give a lot of credit to Chen Yu for this, because it was his genius that allowed us to, at the time, remember, you know, nowadays the field has progressed really quickly, but we were doing these studies back in 2017, 2018, when a lot of the bioinformatics still wasn't fully, you know, these packages weren't fully established or still sort of being used. So what we did here, and this is incorrect, this is not CD45, we sorted based on CX3CR1. I always mean to change that and I never do, so I apologize. But we took light damage retinas from mice and normal retinas. We sorted based on CX3CR1. So these are myeloid cells, including microglia, monocyte-derived cells. And you do a cluster analysis. And what we found was we've gotten, you know, 10 or so different clusters, which I'm showing you here. This is about 10,000 cells that passed, over 10,000 cells that passed QC. And looking here, if you look at the T-SNE plots, most of the normal microglia sit here, which we call MG0 as MG0, just naive. And then in the LD, you can see a complete shift in the microglial populations. You can see all of these sort of, in this cloud, all the microglia sit here pretty much as it's replicating. These are replicating microglia that are slightly different. They're fragmented away from the main cloud. You can see it's amazing. In the steady state, you have really, at this level of resolution, you only have one cluster. And then you hit them with light degeneration, you light model, and all of a sudden, it's like they completely subcluster, which is essentially telling you we have different populations or different states, right?
Danny?
Yeah.
You have two questions in the chat that are related to your light and the RP model, if you want to look at those.
Yeah. Let me see. Maybe you can, do you mind? I
can read it to you. The first question is from Darshan. He's asking, the recruited microglia in light-induced retina and P23H retina are morphologically different. Do you know why?
Okay, so they're different. So remember, the light model is really it's almost like a bomb when off. Can you hear me? It's almost like a bomb went off. Raj, can you hear me? Okay. Okay, it's almost like a bomb went off. So basically phagocytosis, they've got a big fat meal. They look bloated. Whereas in the P23H model, it's more of a progressive degenerative thing. So the cells are not, the photoreceptors are not dying in massive quantities. So they're sort of nibbling and chewing, and so that's why you can see the morphological differences there. Hopefully that answers your question. If not, we can go at it a little bit more at the Q&A session.
The next question is from Tom Ferguson. What happens to the retina if you deplete microglia before light exposure?
We always do that.
Okay, that answers that.
We deplete it essentially 24 hours before.
And then Hank Kaplan, in the RP model, do macrophages infiltrate the inner retina without microglia depletion?
Yes. So that's what we showed with the lineage tracing.
And then Chen, I don't know if this has been typed correctly because you sort of talked about the microglia, but they're asking, did you use PLX compounds to deplete microglia?
So I'm not a fan of the PLX and because there's a lot of problems in terms of no system's perfect, right? The plexicon will kill any macrophage. So if it won't kill it at the monocyte stage, but if the monocyte get recruited in, they become macrophages, it basically depletes them too. The other problem is it's a TK inhibitor. So there's all sorts of issues there with specificity. Of course, you have the VEGF TKs. You have other TKs. I think there's a neutrophil TK. And so, you know, there's a broad range of possible side effects. The CX3CR1 Cre is pretty clean. Of course, we're using diphtheria toxin, and that's a downside. We're also probably hitting all long-lived macrophages in other tissues, which is also a downside. So there's downsides to everything. But no, we haven't done this with the plexicon. Okay. So, and Raj, feel free to interrupt me. I'm just handicapped here with what I can do with talking and seeing screens and questions. Okay, so I'll just rush basically without being too much time. We can determine. See the macrophages here, perivascular macrophages, monocyte-derived macrophages. We know that they're different by genes. That's how we annotate them. For example, the monocyte-derived cells express CCR2, whereas the microglial cells do not. They have high LYZ lysozyme 2. The perivasculars express MRC1 and CD163 and so on. So that's how we can discern them. But at that resolution, we can't see any clustering with normal MGs, normal microglia, which is fascinating. Okay, so how do we, so this is Chen's genius. So how did we have these 10 clusters. How do we find out what's in the subretinal space? So what we did at the time, which was innovative, and now other people are doing it, is this trajectory analysis. And trajectory analysis was used mostly in developmental immune cell or developmental development of cells to look at fate and lineage decisions, not necessarily in the disease context. Essentially what it does is the computer orders the cell. So when you take a snapshot with single cell, it is a snapshot and you're capturing cells at different cell states. So in light degeneration, we're looking at microglia, but clearly we're capturing different cell states of those microglia. And what the computer can do is order those in what's called pseudotime. So just think about it this way. You have magnets, each magnet having a number from 1 to 10, like your kids do. We put it on the refrigerator. You can order it from 1 to 10. You could take them off, mix them up, and then reorder it, right? That's essentially what the computer does. And so you could then hypothesize if it can order it, then, the hypothesis being that this would be the microglia in the subretinal space would be the final state. So you can enter T0 with naive, time 0. And then the last cluster, you would expect that this trajectory would be your subretinal microglia. So you then take these components, you put in T0 as your normal, and then you add these as ask the computer to organize these. Here we left these out because these are macrophages and these are replicating microglia, and their gene expression transcriptome are really different, so it throws it off. And we did 2 trajectories. One was a semi-supervised, and then the second one was a supervised. You can go into the paper if you're interested or ask me questions. How do we get to the supervised? Essentially, it was the one branch that made sense numerically. And then you can see the trajectory of pseudotime from time 0 all the way to time 11 or plus, whatever that means. It's just pseudo, right? And it orders the clusters. And at the end, the last cluster was this small, what we call S, meaning small microglia cluster 3. And so we can hypothesize that this was the final state, which would be the subretinal microglia. And with that cluster, we can then look at differentially expressed genes. One is galectin-3. This is a galectin expressed. There are a whole host of other genes when we compare it to MG0. And you can then plug in these into just a gene ontogeny or gene enrichment pathway. And this was, and I'll get back to this. I know I don't have too much time, but essentially you can see that the upregulated gene pathways, none of them really show a pro-inflammatory neurotoxicity. In fact, they all seem like the microglia were doing something good. Disaccharide bonding, perioxyredoxin, oxyreductase, lipase activity, maybe not good, but certainly not cytotoxic. In fact, it looks like they're responding to cytotoxicity, whereas the downregulated genes were the immune responses. So it seems like these microglia, if these are the subretinal microglia, and I'll show you more data, that they're doing something good, not necessarily pro-inflammatory or bad. Just to show you, then we take those genes and then we can go into the tissue and see where these cells are expressed. So here we're looking at galectin-3, which was really highly differentially expressed, LPL and CD68. And if you look at the GFP, this is CX3CR1, this is GAL3 staining. You can see it beautifully lines here to these subretinal microglia. So if you look at CX3CR1 in the parenchymal tissue, they're green, whereas these in the subretinal space are green and red. So it beautifully labels specifically the subretinal microglia. Of course, there's some end feet. I don't know if this end tissue staining, it could be Mueller cells staining, but suffice it to say that in the context of CX3CR1, only microglia are expressing it in the subretinal space. Say the same thing for LPL. You can see some LPL expression. This is probably vasculature, but you can see specifically with the green cells, they're not LPL positive, only in the subretinal space, and the same thing with CD68. So it looks like we were able to map it back to the subretinal space, and everything again is telling us that microglia have unique access to the subretinal space, whereas the monocyte-derived cells do not in these models. This is the P23H, just to show you with the same markers, we can do the same thing. Here you got in the P23H, I think this is a P60, you can see L-GALS3 staining in the subretinal space here beautifully, whereas the microglia or monocyte-derived cells in the parenchyma are not. And you can see the same thing if you do the RP flat mounts. So what we're showing here, and I'm going to wrap up, this is the last part. I know I'm probably pushing my time limit, but essentially the microglia have unique access to the subretinal space. The monocyte-derived cells do not. And then what the data are suggesting here is that maybe that these microglia are doing something protective. So I'll just show you, go really quick through these slides. How do you know if something's protective? You deplete it, and if you deplete it and the system is exacerbated, you know it's protective. If you deplete it and things are better, then you know it's not. So that's what we did. So we took the light damage model. This is the fundal pictures. You can see in the LD, you can see these focal lesions, these small little dots are representative of microglia. We can show you that. This is the OCT, basically intravital imaging. You can see the outer nuclear layer here. And then that's where the degeneration occurs. In the normal state, you have this beautiful outer nuclear layer, which is not affected. If you look at the DT depletion, you can see that the phenotype is slightly different. Almost looks like it's a bit more exacerbated, let's say. You can see some more hyperreflectivity, suggesting debris. And if you look histopathologically, here are your photoreceptors, that's your ONL. You can see, I'll just skip right to here. In the LD model, without depletion, you can see the photoreceptor thinning. Very few photoreceptors, probably cones. Here's your microglia embedded into the apical RPE. Now look what happens in the depletion state. On quick glance, you'd say, oh, well, it looks like there's more photoreceptors, there's more nuclei. But that's not really true, because if you look at them morphologically, these are all pretty much pycnotic. So the ones that you see here are dead and dying. Essentially, it's debris that was not cleared. And you can also see debris here at the subretinal space. So it seems like the microglia are playing a really important role in clearing the debris. Now, important to realize, the monocyte-derived cells are being spared. So those guys don't seem to be doing any of the clearance, right? And this was the other striking finding here in the light damage model. If you look here at all the microglia DTR lineage trace sticking to the apical side, this is light damage. You can see that the RPE configuration morphologically, these hexagonal shaped cells are beautifully still arranged. If you deplete microglia, it looks like a bomb went off, really. I mean, almost, you could probably argue that there's atrophy clearly, and you can see the dysmorphology is huge. So there's clearly a protective role that these microglia are playing that impacts the RPE. And it also seems to impact the photoreceptor. So here's the P23H setting. You can see this is in P60. These are mice that were not depleted. And these are mice that were depleted. And if you look at the depletion, obviously the photoreceptor layer looks sick, but also you see the subretinal space, what one potentially would call a deposit. You can see this layer here. If you look closely, ultrastructurally, what you would call a deposit is actually completely disorganized microvilli that are sort of removed. And this was statistically significant. I mean, we looked at this in many mice and we saw this repeated. This is not just necessarily a snapshot. So here you have the microglia in the subretinal space that you can see, in the cilia, or excuse me, the microvilli are really ensheathed in the photoreceptor, which is what you'd expect, and this is relatively normal, whereas here it's sort of retracted. So it suggests that there's something happening here with the subretinal microglia, not only in removing the debris, but also in helping interactions possibly between the outer segment and the microvilli. So I'm just going to end with this. I showed you that these recruited into the subretinal space uniquely. Their gene changes, and by the way, this matches Marco Colonna and Ido Amit's work with disease-associated microglia with these gene changes, and they seem to be protecting. And we have lots of new data. We're very close to nailing down this mechanism soon. And if I missed anything from the paper, just to show you the paper, it was published in Immunity in 2019, and I'm always happy to show that we got the cover, which is basically a tree that represents the retina, and you have these different birds which represent microglia, different ecological niches of the tree. And then finally, thank our funding sources, of course, our collaborators who I named, and the postdocs who are on this project, as I named along the way. And of course, Rose Matthew is our incredible lab manager. Without we couldn't run the lab. So I'll stop there.
Danny, I have two questions. One's a quick one, so I'll ask you that first, although I can't see my chat anymore. It was from Tom Ferguson about, do your mice have RD8 or not?
Of course not. We learned that lesson from you, Tom.
Then Ning Shen has two related questions. For the trajectory inference, how do you know SMG is the only end of the trajectory? Are all the subtypes on the same branch? And how many time points did you use for trajectory inference?
So I'm just going to answer this relatively quickly. You'll have to read the paper for all the details, but we had we had basically two branches. One of the branches didn't make sense. And numerically didn't make sense. The number that would end up on that terminal side of the branch numerically didn't make sense. It was just way too high, and some of the genes didn't look right. And we think that cluster, that branch represents the trajectory. We haven't tested this, probably from just the enzymatic digestion. That's what we think. We know that in that last branch, we know because galactin-3 is really highly expressed. Some of these markers are very unique, and those seem to be in the subretinal space. So that's how we can say that. It's not completely finite, of course.
Ning Shen, do you want to ask that last question? Maybe you can ask it better than I can. Or
Sure, thanks. Hi, nice talk. I'm actually from Corona Lab. Yeah, thanks for killing him like a lot of times. But anyway, so I have the question about your single cell RNA-seq data, because to me, your transcriptional, like the pattern, looks quite similar to the Dan microglia signature
Yes,
we
agree.
find in the 5-HCD mouse. However, in that report, Marco published with Ito before I joined the lab, they actually determined that there are two different Dan microglia subclusters. One is TREM2 dependent, the second one is TREM2 independent. And in your gene hits, like I typed it there, like SPP1 and LPL, which is supposed to be TREM2 dependent, but in your case, you don't have the TREM2 hits in that. You don't
So
have the TREM2 upregulation in those, whatever, the subcluster, microglia subcluster 3,
yeah,
but you still have the galactin 3, which also later on, somebody actually published a paper claiming that galactin 3 is actually the TREM2 ligand. Do you know
yes,
that?
yes.
Yes. So I wonder, like, if you are, oh, Marco is here. Maybe he can talk about that. Hey,
The-
Marco.
so the short answer is you're right on target. And Marco and I have been in conversations, and I think it would be awesome to be able to test our theory in Marco's mice with the conventional and the R47 variant. So I think your hypothesis about TREM2 is, I think, spot on. You might not see it enriched so well in our data set. We've looked at it a little bit more carefully. It is. But you have to also realize microglia are not upregulating CD11C either, at least at what we can pick up on the single cell, which is quite different than what Ido and Marco showed. So there's some differences. Clearly, it's the brain versus the retina. But there seems to be an important role for TREM2. We have preliminary data. It's still very, very, very preliminary. But indeed, we are very excited about that angle that you had mentioned without repeating it.
Oh, then since Marco is here, actually, if he's
He's
not here, actually I want to tell him like later. Do you
okay.
think that's actually, if that's the case, if TREM2 plays a role in that, right? Like, do you think it's actually like feasible to apply our anti-TREM2 antibody to see if you can have any like effect on this
Yeah.
retinal regeneration?
Yeah, certainly. But wouldn't we would have to use the humanized mice, right?
Or we have anti-mouse TREM2 as well.
Okay, so we should probably talk.
Right, cool. Yeah.
Thank you.
Thanks, Danny. I just had a last question because you had mentioned you were going to talk about what these microglia might be doing at the bipolar synapses.
Yes.
I think you mentioned it.
So, I want to be careful what I say because this is really outside of my wheelhouse in terms of this, and I see Brian Clark is on, and there are probably other really good developmental and neuroscience scientists that can help us tease it apart. We have some thoughts that maybe it's interacting with negative feedback mechanisms through the amacrine cells. But again, I really don't know. It's something we don't know. We've looked at a little bit with Jeremy K. We know that the number of synapses don't seem to be different. So it's not a quantitative problem. It seems to be more of a qualitative problem, just ruling out that it's not a quantitative problem. We know that developmentally, microglia in the IL-34 mice, they're not affected. So it's not a developmental issue. And the other interesting thing is that ERG that you see, if we age these mice out, it doesn't get worse. It's sort of just stays. It's there, and it's really consistent. So it's very intriguing, and it goes along with the phenomenon that's appreciation that we know that there's some synaptic interactions with microglia. But again, we have people like Sonia Gorel working on this. I mean, these are the people that really, this is the expertise that is needed for this kind of question. And if there's anybody that's intrigued and wants to collaborate, please let me know because, I'm at my limit. We have, we interact with Vadim Arshavsky, but he's more of a photoreceptor degeneration cell biologist. So we don't have the expertise, I don't think, at Duke, at least that I know of.
Dan?
Yes.
Do you have any insight into why the subretinal space, once you deplete the microglia, does not allow macrophages in?
Okay. I get that question all the time. all the time. And you would think that I would probably do experiments to try to figure that out, since there's always that interest. But shamefully, no, we haven't done that. I can tell you intuitively, based on some of the gene changes, I think it's an MMP profile question. It's not a chemotactic signaling question.
Mm-hmm.
Okay, so I think the microglia express certain MMP profile that allows them perhaps to drill through the outer limiting membrane, if that's truly a barrier. The other potential barrier is when you have degenerating and dying photoreceptor cells. So you have this big mess of stuff, and clearly your cell needs to be able to get through that. So
you've not tried interfering with the outer limiting membrane to see whether or not that might, substantiate your idea of passage.
No, I don't. We haven't pursued that angle. It's an interesting one again. Maybe I should, but I can, obviously we're limited by, resources, but it is an interesting question. I wouldn't even know how to interfere the outer limiting membrane just sitting here.
The RD8 model will actually help you. So you probably bred out the RD8 model, but you could actually include that back in, and that will actually have a compromised outer limiting membrane.
Huh.
And they're also
Crumbs one.
They're also pharmacologic way to interrupt it, because that's particularly an issue in terms of trying to get retinal regeneration and extension of the transplanted cells through the outer limiting membrane. So that's another way to do that. But really, very nice presentation.
Thank you, Hank. I'm humbled to hear that from you. Thank you.
You're very kind.
Danny, maybe the last question is from Lynn Hassman. She's asking whether the ERGs improve with resolution of inflammation.
We've not done that experiment. I will tell you this: our data. So we've obviously proceeded with this. What I showed you was our publication from 2019, which is our 1.0, and now we're in version 2.0. We've looked at the ERG responses in P23H with the depletion of microglia, and it is indeed worse. So that fits with our model that microglia are doing some protective. We can't tease apart yet whether microglia are the microglia cleaning up the mess and thereby the absence of mess, which the RPE would naturally want to take up and then cause death to the RPE because they just can't handle that debris. Is that what the microglia are functionally doing in their protective? Is it really just sort of acting like a sink of sorts? And to some extent, that's what's been argued in the plaques in Alzheimer's, right? It's sort of it's protecting the parenchyma from debris of sorts. Is that what it is? Is that all that it is? And of course, it's very hard to tease apart whether it's primarily working on photoreceptors or RPE, because those two are so inextricably linked. You do one to one and sometimes the other, So we haven't been able to tease that apart in our models.
Maybe one more. I have a question from Junction, and I think I'll just sort of change the Junction. Correct me if I'm wrong. He's asking, basically, you've shown that these niches in the retina are important in disease. Is there any similar thing in the brain where the microglia specific niches contribute to brain pathology?
Well, couldn't we argue that, so I'm not a brain person, but couldn't we argue that in the TREM2 or in the Alzheimer's model, that's the case? I mean, we talked about a little bit, if you deplete the TREM, if you don't have TREM2, you don't get that full DAM phenotype. And if you don't have the paper never showed that without the full DAM phenotype, what functionally that would be consequence. It sort of stopped there. But I'm just a simple eye immunologist. I think I will need support from my brain colleagues to be able to really truly answer that question.
Well, Thank you, Danny. This was terrific and great discussion. Thank you again. Sorry it has to be virtual, but we really enjoyed your talk.
Thank you.
For attending.